If you ask the general adult population, about 20% will tell you they are chronic procrastinators. But step onto a university campus, and that number skyrockets to 70%. Procrastination in higher education emerges when personal stress, self-doubt, and anxiety about performance outweigh a student's self-control and motivation. By putting off assignments until the last possible moment, students force themselves into highly inefficient cramming sessions. They attempt to condense weeks of learning into a single high-stress window, disrupting the sleep cycles required for memory processing. This creates a behavioral loop that reduces the efficiency of the biological mechanisms the brain uses to process and store new information. Long-term memory retention ignores the total hours you spend staring at a textbook. It relies on a precise two-part biological equation. The first phase is cognitive encoding. This is the active acquisition, filtering, and structuring of data that happens while you are awake. The second phase is physiological stabilization. This is the offline processing that occurs during sleep, where those newly formed neural traces are secured against forgetting. To understand how to optimize this 24-hour cycle, we can look at empirical data from medical student performance and clinical sleep trials. Mastering complex material requires successfully executing both halves of this biological sequence every day. When students cram, they often default to passive reading. They scan highlighted text or reread notes, methods that yield very little long-term retention. According to cognitive load theory, learning requires deliberately retrieving information from your memory. Instead of passively absorbing facts, the brain must actively reconstruct them. This chart plots retention over time. The steep drop shows the rapid decay of passive review. But when medical students use spaced active recall, distributing their self-testing over time, they see a 30 to 50% improvement in information retention. Dense, technical fields demand this kind of mental friction. The act of retrieving data strengthens the neural pathways holding that information together. Active retrieval ensures a newly formed memory trace is robust enough to survive the cognitive interference encountered throughout the rest of the waking day. Once encoded, a memory enters the second phase. Research since the 1920s suggests sleep protects these fragile memories from the continuous input of waking life. This creates a practical question for a student's schedule. If you study in the morning and face 12 hours of waking interference before finally going to bed, do you lose that protective benefit? This diagram outlines a study by Gasparello and colleagues designed to test that exact time dependency using 48 participants. The orange track represents the sleep first group. They encoded new data at 9 p.m. and immediately went to sleep. The purple track shows the wake first group. They encoded the exact same data at 9 a.m. and spent the next 12 hours awake, exposed to the normal cognitive interference of an active day. Both groups performed a declarative task, memorizing specific geographical facts connected to maps, location photos, and short audio clips. This parallel design isolates the variable of waking interference to see how it affects the memory traces before they can be stabilized by sleep. This graph tracks their performance. The T0 column on the left shows the immediate recall test taken right after the study session. Both groups start with a similar high baseline of accuracy. Moving 12 hours forward to the T1 column, we see the performance of the sleep first group. After a full night of rest, they show only a modest decline in recall. Sleeping shortly after encoding allowed for efficient memory stabilization. We contrast that with the purple wake first group over the same 12-hour period, showing a much steeper drop in performance. 12 hours of constant waking interference caused significant decay in their ability to retain the facts they had just learned. At the 12-hour mark, sleeping immediately after learning provides a clear advantage for retaining facts. To see the full cycle, we look at the final 24-hour test at T2. By this point, the wake first group has finally slept, and the sleep first group has faced their own 12 hours of waking interference. The results show that at the 24-hour mark, both groups converge to almost the exact same retention level. Even though it was delayed, the wake first group's sleep managed to stabilize their remaining memory traces. The sleep first group lost their early advantage because they eventually encountered 12 hours of waking interference after they woke up. The data suggests that the sheer occurrence of a full sleep cycle within 24 hours is what stabilizes memory. regardless of whether that sleep happens immediately or hours later. We can now evaluate procrastination through this biological lens. Delaying work leads to massed practice, or cramming, where a high volume of data is processed in a single, high-stress window. Cramming typically relies on passive review, which misses the 30 to 50% retention gains provided by spaced, active recall. Pushing this workload into the early morning hours often results in a shorter sleep cycle. Shortening that sleep period limits the window for offline stabilization. Without a full cycle, those new memory traces remain more vulnerable to the interference of the next waking day. Chronic procrastination results in a less efficient learning process, preventing the brain from reaching its full retention potential. The empirical evidence suggests a specific 24-hour learning protocol. Phase 1 requires high-intensity active recall sessions, separated by intervals of space, to build strong neural traces. Phase 2 requires a full sleep cycle within 24 hours of encoding. This offline processing is essential for long-term retention, regardless of when you studied during the day. While you cannot avoid waking interference, securing proper sleep ensures that your baseline memory survives to the next day. Academic success depends on treating focused study and restorative sleep as two halves of a single biological equation.